Welcome back again, friends. Welcome back to another video session from this channel. So in this video session we will be studying regarding the alternative strategy through which the RNA editing mechanism it is successfully organized in case of various living cells. And here it is nothing but what? Guide RNA mediated nucleotide insertion or deletion. Is it clear? So here, prior to the explanation of guide RNA mediated nucleotide insertion or deletion, at first let us have a look in which particular living organism for the very first time the guide RNA mediated nucleotide insertion or deletion it was identified. Is it clear? So here for the very first time Rob Bennett and as well as his colleagues they have identified the guide RNA mediated nucleotide insertion or deletion in a specific protozoan which is nothing but what? Trypanosoma brucei. Is it clear? So let me write it on the board. Who is he? Rob Bennett. Rob Bennett and colleagues, they have identified the guide RNA mediated nucleotide insertion or deletion in a specific protozoan which is nothing but what? Trypanosoma brucei. It was observed in a protozoan which is nothing but Trypanosoma brucei. Is it clear? So here in case of Trypanosoma brucei, the cellular mass of DNA, here it is termed as what? Kinetoplast. Cellular mass of DNA, it is termed as what? Kinetoplast. Is it clear? So here what is kinetoplast? Here kinetoplast, here it is nothing but what? It is an aggregation of maxi circle and as well as mini circle. Is it clear? So let me print it on the board. Maxi circle and as well as mini circle. Maxi circle and as well as mini circle. So here it is nothing but what? Maxi circle, right? And here it is nothing but what? Mini circle. Is it clear? So here once when the maxi circle and as well as mini circle, when they undergo aggregation, it leads to the formation of what? Kinetoplast. So here within the maxi circle and as well as mini circle there is an availability of 30 mitochondrial genes. Is it clear? So here out of 30 mitochondrial genes, 12 of the mitochondrial genes they are under cryptogenic condition. Is it clear? 12 mitochondrial genes in case of kinetoplast, they are under cryptogenic condition. Cryptogenic condition. Is it clear? So here what is the meaning of cryptogenes? So here the specific gene it is capable to induce the expression of mRNA, but the specific mRNA during the translation mechanism it is unable to induce the synthesis of protein. Is it clear? That means the specific genes they are under inactive conformation or they are under non-active conformation. Is it clear? So here let us consider the specific gene. Here the gene during the transcription mechanism it is capable to induce mRNA. But the specific mRNA during the translation mechanism, it is unable to induce the synthesis of protein. So here the cryptogenes, they are non-active genes or inactive genes. Is it clear? But here, by taking the advantage of guide RNA, once when the specific guide RNA, when it comes into the picture, now the specific mRNA, after the successful synthesis of mRNA during the transcription mechanism, now the specific mRNA, it undergoes post-transcriptional modification. What it is? Post-transcriptional modification. Right? Is it clear? So here the post-transcriptional modification, it is nothing but what? RNA editing, guide RNA mediated editing. So here once when the specific guide RNA, when it comes into the picture and once when it inserts certain uridine residues into the specific mRNA, under such condition, now the specific edited transcript, when it participates in the translation mechanism, it is capable to induce the synthesis of protein. Is it clear? So initially the specific mRNA, it is unable to induce the synthesis of protein, but once when the edited transcript, when it participates in the translation mechanism, it is capable to induce the synthesis of protein. It is because of what? Guide RNA mediated nucleotide insertion or deletion. Is it clear? If we consider the structural components of guide RNA, structural components of guide RNA. Is it clear? So here basically the specific guide RNA, it is a non-coding RNA. So let me write it on the board. It is a non-coding RNA. And here it consists of three domains. What are those? One is anchoring domain. Anchoring domain. Is it clear? And the other one is editing domain. Editing domain. And the other one is tail domain. Tail domain, right? Is it clear? And here in order to organize the guide RNA mediated nucleotide insertion or deletion, the specific mechanism or the specific process, it takes the advantage of a specific complex which is nothing but what? 20S editosome. Is it clear? What it is? 20S editosome. So here the 20S editosome, it is an association of three different enzymes. What are those? Endonuclease, exonuclease, and the other one is ligase. By taking the advantage of all these enzymes, here the guide RNA mediated nucleotide insertion or deletion, it is successfully organized. Is it clear? So here the guide RNA, it acts like a template. So by depending on the information which is present in case of the guide RNA, insertion or deletion of nucleotides takes place. Is it clear? So let me write it on the board. Guide RNA acts like a template. Acts like template. So here for your better understanding of guide RNA mediated nucleotide insertion or deletion, let me give you one common example. Is it clear? So here if we consider one specific mRNA, So here the nucleotides are nothing but what? G, A, G, U, A, U, C, C, U. And here it is nothing but what? The 3' end. Is it clear? Here it is nothing but what? The mRNA. So here the 3' terminus of the mRNA, it base pairs with 5' end of the guide RNA. It is the 5' end, right? Is it clear? So here the complementarity of U is A. U is A. And the complementarity of C is G. Is it clear? And the complementarity of C is, again it is G. A, U, A, G is C. So here the complementarity of A is, in general it is U in case of RNA, but here there is an availability of one specific mismatched nucleotide, and it is nothing but what? A and A. And here there is an availability of some more other nucleotides at the 3' end. Is it clear? So here when there is an availability of mismatch, under such condition, in order to cleave the mismatch here, one specific enzyme which is nothing but what? Endonuclease enzyme, it comes into the picture. Is it clear? Endonuclease. As I have already mentioned you, in order to organize the guide RNA mediated nucleotide insertion or deletion, there is a requirement of a specific complex which is nothing but what? 20S editosome. So here the 20S editosome, it is composed of endonuclease. So here the endonuclease, it is responsible for the cleavage here. Is it clear? So let me print it here. 5' end GA. So here the cleavage takes place, right? A, U, C, C, U. So here it is nothing but the guide RNA. The complementarity of U is A, C is G, G, A, U, A, C. And here there is an availability of A and A, right? So here, once when there is an availability of A and A, in case of guide RNA, the complementarity of adenine is, it is nothing but what? Uracil or the uridine. Is it clear? So here for the successful insertion of the uridine nucleotides into the specific mRNA, here the specific mechanism, it takes the advantage of a specific enzyme which is nothing but what? Terminal uridyl nucleotidyl transferase. Is it clear? So by taking the advantage of terminal uridyl nucleotidyl transferase, here the successful insertion of the uridine nucleotides into the specific mRNA takes place. So as I have already mentioned you, by simply depending on the information which is present in case of the guide RNA, insertion or deletion of nucleotide takes place. So here once when there is an availability of adenine in case of the guide RNA, so here the complementarity of A is, it is nothing but what? U. So here for the successful insertion of uridine nucleotide into the specific mRNA, the specific mechanism, it takes the advantage of an enzyme which is nothing but TATase. So here TATase is nothing but what? Terminal uridyl nucleotidyl transferase. And here the precursor for the successful insertion of uridine nucleotide into the specific mRNA, it is nothing but what? UTP. Is it clear? So let me write it here. UTP acts as a precursor. And the complementarity of A is U and the complementarity of G is C here. And here it is nothing but what? The 3' end. So here it is nothing but what? The guide RNA. Is it clear? So here after the successful insertion of the uridine nucleotide, then after one more other enzyme it comes into the picture, and here it is nothing but what? Ligase. So here ligase is responsible for inducing the ligation. Is it clear? Ligase enzyme. It induces ligation. Is it clear? So once when the ligase enzyme, when it is capable to induce the ligation, so with this, the guide RNA mediated nucleotide insertion or deletion, it is successfully organized. So here you may get a small clarification. Why always the guide RNA, it targets only the uridine nucleotide instead of targeting any other nucleotide like A, G and C? Why the specific guide RNA, why it only targets the uridine nucleotide? So here, as far as my knowledge was concerned, the answer to this question is, in comparison to the other nucleotides like A, G and C, uridine nucleotide, it is very less stable. Is it clear? So that the specific uridine nucleotide, it is capable to cleave so easily, or it is capable to insert so easily into the mRNA. So the reason why the specific guide RNA most commonly targets the uridine nucleotide for the successful insertion and as well as deletion into the mRNA. Is it clear? So here this is regarding guide RNA mediated nucleotide insertion or deletion, which is most commonly observed in case of certain protozoans like trypanosome of brucei. Is it clear? So here I hope that this video will help you a lot. So if you like this video, just hit the like button and share it to your friends. And I remember you people to subscribe my channel for getting more and more videos what I make day to day. Thank you.